Hello AP Bio students, this is Mr. Laudermith. I want to take a couple minutes and talk about testable hypotheses of evolution. In other words, if we are going to say that Darwin's theory of evolution by natural selection is a true scientific theory, then we ought to be able to use the principles of natural selection to test hypotheses, to find answers to scientific questions, to solve scientific problems. Just like we can use Newton's theory of gravity to answer questions about the movement of planets in the solar system, we should be able to use Darwin's theory to answer questions about why populations exist the way they do in the natural world. So let's look at a couple examples, starting with a famous one, the peppered moths of England. In the forests of England, there's always been two varieties of peppered moth, a light variety, this one on top here, and a dark variety. They are just two separate genetic variants, two phenotypes, if you will, of the same species. These are both the same species of moth. They could breed together, but every moth that hatches is either a light color or a dark color, depending on the genes it inherited from its parents. Now, it probably doesn't surprise you that when trees are dark, the dark-colored moths blend in better and the light ones stand out. And if the trees are more light color, the light-colored moths blend in better and the dark ones stand out. Nothing surprising there. So let's look at some information that was gathered over a period of about 150 years. Starting in the 1840s, sampling of moths in England's forests found that about 95% were the light variety, 5% the dark. 50 years later, 98% are dark and only 2% are light. Interesting reversal there. And about 100 years after that, by 1995, we've got 19% dark, 81% light. Well, we can ask a question about this. Why did the percentages of dark and light moths change so dramatically over this 150-year period? I bet it's not going to surprise you when I say that the answer is natural selection. Well, a little bit of artificial selection thrown in, as you're about to find out. In the early 1800s, we have a pre-industrial England. This is before the Industrial Revolution. Not much pollution. The trees have this light-colored lichen growing on them. The bark is light-colored. By the late 1800s, England is in the full swing of the Industrial Revolution. All of its factories are run on coal power. When you burn coal, you send tons of dark soot up into the air, lots and lots of air pollution. It falls back down on the trees. It covers the bark of the trees. Now the trees are all dark in color. By the end of the 20th century, we have pollution controls in place. We have clean air laws. As a result, we don't have factories churning out all this coal soot anymore. The trees have been restored back to their original light color. Well, if we look then and see what was happening in England in each of these periods of time, now we can understand why the moths changed. In the early 1800s, if trees are light in color, the light-colored moths would have the advantage. They would be less likely to be spotted by predators and have a greater chance of surviving and reproducing more light-colored moths in the next generation. When the trees became darkened by pollution, the dark moths could blend in better. They're the ones who survived more often and created more offspring, who are also dark in color. By the end of the 20th century, the trees are back to being lighter colored. Now it's advantageous again to be a light-colored moth. They are the ones more likely to survive predators because they can hide better. They're the ones who get to reproduce. We call this particular example of natural selection in action industrial melanism, change in coloration brought on by human industry. Okay, let's look at another example. This one involving Darwin's finches. You probably thought you knew everything there was to know about Darwin's finches already, but it turns out that since the 1970s, Rosemary and Peter Grant have visited the Galapagos Islands, and they've looked at a group of seed-eating finches called the medium ground finch. And every year they capture the finches alive. They measure the size and depth of their beaks, but they also then look at the amount of rainfall in that particular year. And they've been able to correlate those two measurements, beak depth in offspring and the amount of moisture. It turns out that in dry years with less rainfall, the plants on the Galapagos tend to produce thicker shelled seeds. Well, if the seeds have thicker shells, to crack them open, you're probably going to need a bigger beak. Unsurprisingly, in those years, it's only the really large, deep-beaked finches that get enough food and survive, and they pass on that bigger beak to their offspring. So you can see in dry years, we have kind of a peak in beak depth. We see more offspring with bigger beaks. In years that are wetter, with more rainfall, the plants tend to produce seeds with thinner shells. Having a smaller beak is no longer a disadvantage. Having a smaller beak allows you to get plenty of food, and in those years we tend to see a decline in the average size of beak depth because, again, having a really big beak is no longer super advantageous, and it's not a disadvantage to have a smaller beak. So we would expect that the average beak depth would get smaller in the wet years. So again, correlation between beak size and shape and food source, but now tied to an environmental factor. So here we can use the theory of natural selection to explain how the amount of rainfall influences a particular trait in a population of birds. Okay, natural selection in action. I've mentioned this in another video. We can talk about insect resistance to pesticides or bacteria resistance to antibiotics. In both cases, some individuals in the population will have a genetic trait. They will possess a gene making them resistant to that particular toxin. Then, when the insecticide or the antibiotic is used, it does not kill all individuals. Those who have the resistance gene survive. They're the ones who get to reproduce with each other. Their offspring all inherit the same resistance gene. As a result, the next generation has a greater percentage of individuals who are resistant. And as long as humans keep applying the same insecticide or the same antibiotic, killing off the ones who are not resistant, the percentage of the population that is resistant is just going to keep going up and up, until you have populations that are basically entirely resistant to the insecticide or to the antibiotic. It does not harm them in the least anymore. Now, we understand this principle from a Darwinian perspective, but we might then follow up with a question, how is this resistance actually affecting the organisms on a cellular level? Well, it turns out that in insects who possess a resistance gene, their particular protein receptors in their cell membranes that would normally allow the pesticide molecules to enter the cell, they now have a different shape to them, and they actually block the pesticide molecules. So the resistance gene actually leads to a differently shaped cell membrane protein, which now does not allow the pesticide molecule in, and that is why those individuals are able to survive and reproduce. So here we can look at Darwin's theory of natural selection, but actually go deeper with it to the cellular or even the molecular level, and we can see how the resistance is actually affecting the specific traits of these organisms. Okay, let's look at one more example. How about humans? Is there anything Darwin's theory of natural selection can tell us about changes in human populations? Well, in fact, it can. But we're going to look at a trait here that affects the entire human species. Now, it's long been known, of course, that chimpanzees are genetically our closest relatives. About 96% of our DNA is identical to a chimp's DNA. Now, that's interesting in and of itself, but there's also a significant difference in our chromosomes. Turns out that if you look at the great apes, the orangutan, 48 chromosomes. The gorilla, 48 chromosomes. The chimpanzee, whom you'll recall is our closest genetic relative, 96% of the DNA is the same, 48 chromosomes. And then the human, 46 chromosomes. I bet you remembered that from your last biology class. Now wait a second. If we are so genetically similar to these other species, especially the chimpanzee, shouldn't we also have 48 chromosomes? What could be a possible explanation for that? Well, I guess you could say, well, sometime in the past, human ancestors just lost a pair of chromosomes. That's a possible explanation, except that, remember, there are really no such thing as chromosomes that do nothing. Chromosomes carry the genes. Each pair carries many thousands of genes needed to perform cellular functions. So it's probably unlikely that human ancestors just lost 2 chromosomes somewhere along the way and were none the worse for it. So is there another hypothesis we could test regarding this change in chromosome number? Well, if these organisms share a common ancestor, then is there evidence somewhere in our chromosomes to account for this change in chromosome number? Maybe it's not a lost pair. Could it just be that there's a fused pair? In other words, human ancestor, an ancestor as well of the chimpanzee, gorilla, orangutan, had 48 chromosomes, 24 pairs. Could one of those pairs have fused, have linked together with another pair, reducing the number down to 23? If you think about that for a second, if that's what happened, we didn't lose any genes. All the genes are still there. It's just what were 2 separate chromosomes have now linked together to form 1. That would explain the drop in a pair and the loss of 2 chromosomes, because it's now not really a loss. All the genetic material is still there. We just don't think of it as being 2 separate chromosomes. So there's the 2 chromosomes in the ancestor. What we're hypothesizing is that at some point in the past in a human ancestor, those 2 separate chromosomes fused together at these endpoints called the telomeres. If that's what happened, we should be able to look inside a human cell nucleus and find a longer chromosome where there are 2 of these telomere regions right next to each other. And if we can find that, that would be good evidence to back up our hypothesis of this fused chromosome in the ancestor. This is a testable hypothesis. We're saying this is what we should find inside the nucleus of a human cell. Let's go and see if we can find it. Testable hypothesis, scientific theory. Well, biologists went looking. 2005, guess what they found? Human chromosome number 2, which is our second longest chromosome, they did in fact see that there were 2 of these end regions, 2 of these telomere regions right next to each other. The only time we'd ever expect to see that is if chromosome 2 was originally 2 separate chromosomes that became linked together in the past. So, we could even take it a step further, and these scientists did. Which 2 chromosomes became fused together? Well, we went looking in the chimpanzee genome at chimpanzee chromosomes, and they found 2 there that if you put them together end to end, would basically make a chromosome the same size as human chromosome number 2. So we knew what we had to find to back up our hypothesis. We found it. It was a successful test of the hypothesis. Chromosome 2, we can say, is now unique to human lineage because unlike other great apes, it is the result of 2 separate chromosomes that became fused together in the past, giving humans 46 chromosomes, 23 pairs, not 24. So I hope you've enjoyed hearing some of these examples of how we can use Darwin's theory of evolution to answer scientific questions that affect humans, but also that involve human evolution itself. Thanks for listening.